Hello guys, I am Shubham from ShubhVed Tutorials. In the previous video, we saw what are linked genes, right, and what are unlinked genes. So now we will see how these linked genes are inherited. Okay, means passed on to the offspring, right? So now in linked genes, see this is the F1 offspring which is having this type of genotype, right? Now this is the F1 that is having different genes on same chromosome. These are linked genes, as we saw. So okay, linked genes. Okay, so these are linked genes, and right, then they will segregate, they will form, right? So these are the gametes formed. Okay, so basically here we can see only parental phenotypes, right? Only parental phenotypes, that is capital AB and small ab. Only we can see these types of gametes, right? So there are no recombinant gametes. Okay, as we saw, recombinant means capital A with small b and small a with capital B. These are recombinants that we does not see. Okay, so this type of linkage, this type of inheritance is known as complete linkage. Complete linkage, in which there are no recombinants seen, only parental genotypes we can see. Okay, in the next case, same is the condition, right? Same is the condition. Here also same condition. Sorry. Okay, okay, got it. Now what happens? There is a cross between this thing. Cross. Okay, cross. So now what happens? These are the same, as well as these are the same. Between these two chromosomes, so here there occurs the exchange. Okay, here there occurs the exchange. So basically, what will happen? See, this will take some part of red, some part of red, and the other one will take some part of blue. Right? So this is known as crossing over. Okay. So instead of instead of capital A, there will be small a there. Okay. So this will be small a, and here capital B as it is. Here will be capital A, and here small b as it is. Okay, and then segregation. Segregation again. Alright. So this type of genotypes you can see of gametes. See, this is AB, this is small b, again capital A, small b, and here small b. Right? So here we can see parental, right? So these two are parental, and this the two small these are recombinants, and this parental. Okay. So what we can see, 50% individuals, 50% parental, and 50% recombinant. Right? So parental and recombinant. So now this type of linkage or inheritance is known as incomplete linkage. Incomplete linkage, in which we can see recombinant offsprings, recombinant gametes too. Okay, in complete linkage there is no crossing over does not occur, and thus recombinants are not produced. Right? But in this type, crossing over occurs and recombinants are produced. Right? So for this recombination, okay, what we can see, due to crossing over, recombinants are produced, or in different, if the genes are located on different chromosome, then due to independent assortment, we will see recombinants are produced. So with the help of this thing, recombination, there is a percentage of recombination, that is 0 percentage to 50 percentage. Right? So 0% indicates that there is no crossing over. 0% means no crossing over. Means we can say that complete linkage. We can see complete linkage. And 50% means crossing over has occurred. Crossing over has occurred. Means 50%. Okay. So when we will see 0%, in the case of complete linkage, okay, complete linkage, complete linkage, right? And this is crossing over incomplete linkage. Incomplete linkage. Got it? So this is the case. Okay. So when, see, suppose this is a chromosome, again we will draw a chromosome, and if the genes are close to each other, then there will be no crossing over. If they are close, okay, then they will come as it is. Right? Okay. So this will also come in here. No crossing over means they are located too close, so that they are not separated from each other. So in this case, we will see no crossing over. If the genes are located on the same chromosome, but they are located too close to each other, so that they cannot be separated. But if the case is, now the next case we will see, If the genes are located far away from each other, then they can get separated. Okay, they can get separated. Crossing over can separate these two genes, right? And here crossing over, if the crossing over occur, then also these genes will not be separated because they are too close. Because they will be inherited together only in the first case. And the second, they can get separated because they are apart from each other. Okay, so 0% and 50%. So less than 50%. That is one criteria now when we will see the percentage. Suppose lesser than 50, equal to 50, and greater than 50. Right? Lesser than 50 means genes are close to each other. Okay, no crossing over, or crossing over of will occur, but the genes are close to each other. I will just write here genes close. Okay, genes close. Right? 50% means recombination can be occurred. Equal to 50% means crossing over has occurred, and what we can see, incomplete linkage. Genes are in this both the cases. We can see that equal or greater than. Okay, equal or greater than 50. Equal or greater than 50. In this, we can see genes are far apart. Genes far apart, or we can say that unlinked genes means genes on two different chromosomes. Right? If they are linked, then they will be far apart. Okay, and if they are unlinked, then they will be greater than 50. Okay, so that's it. These are the types of linkage. Linkage means inheritance of how linked genes will be inherited. So that's it for this video. I hope this video helps you. Thank you guys for watching this video. If you like it, please share it. Don't forget to subscribe and press the bell icon to get notified whenever new video is out. Also follow us on all our social media pages, and you can join us on Telegram and stay connected for the latest updates. All links mentioned in the description section.